Hello and welcome to a webinar on non-sterile product testing. My name is John. I'm the department manager here at Nielsen Labs in the microbiology department. In our department we test non-sterile products, for example pharmaceuticals, raw materials, water, ingredients, finished products, for lot release, environmental monitoring, incubation of air test healthcare items, and cosmetics. Pretty much anything that you find at the store in the healthcare aisle we test. And we also test some non-sterile tissue products. Today I'm going to be covering product type, sterile versus non-sterile products, a bit about pharmaceutical microbiology basics and history, microbial control strategy and risk management, non-sterile standards and testing, and setting acceptance criteria for non-sterile products. So looking at the type of products, which products have more risk. Sterile products are sterile for a reason. There needs to be a complete absence of organism because they're going to be used usually in the bloodstream or in the body like a medical device or an injection. And they are aseptically manufactured, which has a higher risk and extremely high control during manufacturing, or they're manufactured and then terminally sterilized, which has a bit of a lower risk. When looking at non-sterile products, they have a moderate to low risk, which is why they're able to be non-sterile. They're able to they have organisms in the product, but unlike sterile products where it's very cut and dry the requirements, non-sterile products require a risk assessment where acceptance criteria is set. Generally, there's a bioburden that's set for the products to see have a limit for how many organisms can be recovered, and also absence of objectionable organisms is an FDA requirement, and we'll talk a bit about that. First, we want to look at a risk hierarchy for pharmaceutical products. As we covered, sterile products on the very top are at the highest risk, going down through non-sterile nasal sprays, topical products, down through to oral tablets and powdered filled capsules. Those type of products are the least risk because generally there's very little to no water content, and that causes the risk to the user to be extremely low. If a healthy human being takes some medicine, takes a pill for a headache, there's a very low chance of any risk. But on the other side, if a immunocompromised patient was taking an oral tablet, risk may be a little bit higher. So requirements for non-sterile product testing are to be free of objectionable organisms. To go through some definitions, a pathogen is a microorganism that causes disease in the host. So here we're going to be looking at bacteria and fungi, and covering two types of pathogens. First, obligate pathogens are pathogens that need a host to reproduce. They can survive outside of the host, but they cannot proliferate and reproduce. A disease is caused by transmitting one from one host to the other. Examples of this are STDs, tuberculosis. Next is an opportunistic pathogen. These are pathogens that are organisms that are everywhere on our skin, in our normal environment. They usually don't cause problems unless there's a change in that environment. For example, yeast infections yeast is able to, excuse me, yeast is able to grow because the environment changed and the bacteria were not there to have an even environment. But given the right circumstances, any organism can be invasive and possibly lethal. So what exactly is an objectionable organism? The answer really depends, and it's on the product, how it's used, who uses it, and that's determined through a risk analysis, which we'll cover in a bit. Before we cover that, I want to cover at least a short history to see, to cover how we got to where we are with regulations. There was a realization that these pharmaceutical products, based on how they were administered, caused problems. So here we have eye drops, ophthalmic dosage forms. Previously, before there was much regulation, there was little control. Eye drops are generally a multi-dose product, so they're used multiple times. And there was no limit or volume, limit on the volume. So there could be a large glass jar that would be used over and over, which can cause recontamination when they're used. And there weren't many regulations on preservatives. Mostly they were thought to be have preservatives just by low nutrients, but Pseudomonas aeruginosa is able to grow in that environment, and that caused eye infections. And if there was a cork closure, that's pretty much contaminating the sample with mold spores. So there was an overhaul of the requirements. The requirement was to have eye drops as sterile, have lower volumes so that when they're used, they can't be used over a long period of time. And there must be a broader spectrum of preservatives for the multi-use that target specific organisms and, in this case, make the product pretty much sterile every time it's used. Next, looking at dosage forms. Again, that's a multi-use product. There was some preservatives, usually alcohol is used, which isn't a great preservative, especially since they could be in very low percentages. There was lots of problems with food contamination, food poisoning, and a lot of this was caused by the raw materials. Since a lot of the pharmaceutical products were, the active ingredients came from roots or flowers, the raw materials brought in salmonella because the natural ingredients were in the dirt. In the dirt there's manure, and in manure there's enteric organisms. So in the 60s there was implementation of a requirement of a low bioburden, which goes back to that acceptance criteria, and an absence of enteric pathogens. So that's an example of an objectionable organism for oral dose products, because those specific organisms would be opportunistic in the way it's used, so since it's swallowed. Through the regulations and learning how the non-sterile products affect the users, there was a development of methods to ensure quality products and microbial control during manufacturing. So monitoring is important, looking at all the products, having raw materials tested, finished products tested, monitoring the environment, use of preservatives for multi-dose products, aseptic manufacturing and sterilization for the right areas, and packaging to ensure that the container closure system is appropriate for the product. Looking at the regulations, the GMPs require written procedures to prevent objectionable organisms for non-sterile products. And through the prevention, to have a prevention system, microbial control strategy and risk management are really important. In the past, there hasn't been much guidance for non-sterile manufacturers. It was usually based on sterile manufacturing, and non-sterile manufacturers were using some of those systems. Recently, the USP's released a draft version of USP 1115, which uses a risk-based approach to control bioburden during manufacturing non-sterile products. And we'll cover some of that information here. So it's important to identify the sources of contamination in the control of organisms. Looking at facilities, is the facility built correctly, organized correctly to allow products to be produced without contamination? The environment, ensuring that the environment is controlled in areas that are needed. Equipment, cleaning procedures for the equipment, and if they need to be replaced. People is really important, because going back to pathogens, for non-sterile products, we're mostly concerned with opportunistic pathogens, which again is part of the normal microbiome for humans, and people could be one of the largest contamination issues during the manufacturing. Container closures, as we covered, and raw materials ingredients is very important to look at for control of contamination. As we saw with the oral dose products, organic materials are going to have much of a higher risk than using synthetic materials. Through control, sterilization may be appropriate for water or those organic materials before they're put in the product, even for non-sterile products, depending on what the material is. Generally, with non-sterile products, toxins aren't quite of a issue. However, I have this here just to lay out that it's important to audit your suppliers for non-sterile products. You don't want the supplier to have an environment that would be favorable for organisms to grow. Next, for materials, API, are they synthetic, fermentation, or cell culture? Do they contain protein or peptide active drugs that would have nutrients available for organisms? And how do you control those organisms? Water activity, if the product can be dry, that's a way of contamination control for multi-dose products, preservatives, and packaging. Storage conditions, lower temperatures will reduce or prevent proliferation in the product. So next, we will look at testing and release for distribution. The GLPs require microbiological testing before use, and appropriate lab testing to make sure the products are free of objectionable organisms. So I'll cover 3 tests that are outlined in the USP. First is the USP 61, which is the Microbial Examination of Non-Sterile Products, Microbial Enumeration. The purpose of this test is get to the overall bioburden, or the number of CFU that can be recovered during the test, looking specifically at bacteria and mold and yeast. This is a quantitative test, so we're going to get an actual number. For example, 10 CFU per mil. The testing can be used to monitor in-process testing with raw materials or lot release testing. And this is a harmonized test, so the same methods are used for the USP, EP, and JP. During the test, the sample is prepared in a sterile broth or buffer solution. And during the testing, the sample preparation is placed onto agar plates. For total robot counts, it's a general agar that will grow most organisms that we're looking for. For the yeast and mold counts, there's a lower pH, which promotes growth of molds and yeasts. And at the bottom there, I have growth function as an FDA Focus. It's always important, even not just in this case for this test, but environmental plates or any media used, to have growth promotions to show that the agar actually is able to grow the organisms that you need to for each lot. USP outlines the requirements for sampling and sampling size. Generally 10 grams, milliliters, or patches are recommended, unless there's a smaller batch, for example, a API that only has 100 grams. 10 grams is going to be far too much to actually test. But a different amount can be used as long as there's a justification. And the sampling must represent the entire lot tested. And it's always good to train to get a baseline and increase sampling if you reach a action or alert level. The requirements for GLP for a sampling scheme, what and where to sample, beginning, middle, and top, middle, bottom are generally ways to get testing done that represents the entire lot. In the sampling scheme, you should have the amount of sample needed. You may need to take out 100 mils or grams to get a representative amount for that lot. The scheme should outline sampling, how often it should be performed, and have very specific methods and procedures for training precautions to avoid contamination, and if equipment needs to be sterilized when cleaning. Also, strict requirements for sample labeling, handling, and storage. If the products need to be placed into a lower temperature or shipped during a temperature, that should be metered during the shipping. So for the preparation, again, the sample is prepared in the non-sterile broth, or excuse me, the sterile broth, and the goal is to get a homogeneous mixture to ensure that a representative amount of the sample is tested. So for water-soluble products, not too much of a big deal since we're using water-based media. But for fatty products, gelatinous products, aerosol materials, there are some methods to get those to dissolve, or, for example, with transdermal patches, more of a extraction is performed. There's 3 methods outlined in the USP 61. First is filtration. That's where the sample preparation is placed, it is vacuumed through a filter that catches the organisms, and then the filter is placed on the appropriate augers. Or plates can be performed, and that's where a small amount of the sample preparation is placed into a sterile petri dish, and a molten agar is placed into the dish and it's mixed. But if the sample can't If the sample is very gritty and has larger chunks, say like white powder, it doesn't it isn't water soluble. Surface spread method may be more appropriate, so you can actually count the colonies on the top of the surface. And that's done by taking a small amount and spreading it on top of a agar plate that's already poured. Once that's done, the incubation is done, the plates are counted, and the results are recorded, calculated based on the dilution performed. With the testing, it's very important to have a suitability performed. A suitability demonstrates that the test actually is valid and shows that if there are any inhibitory properties of the sample, that organisms can be recovered during the test. So during the testing of with suitabilities, the sample is actually inoculated with a set number of organisms and types of organisms ran through the test, and growth has to be recovered compared to a positive control, a 50 to 200% recovery. And again, that shows that the testing is valid and that you're not getting false negatives. With USP testing, with any test, there are limitations. So not all organisms present in the product will grow during this testing. They may be viable but not culturable. Generally in products, non-sterile products, they're going to be, the organisms are going to be very stressed, and putting them in a high nutrient area media may not be able to revive them. During the suitability, a very low number of organisms are inoculated, and that helps at least show that a low number of organisms can be recovered. And organisms and microorganisms in samples are not homogeneous. So when you're taking samples, it's important to make sure you get the representative lot, because organisms may be only on the bottom, maybe floating on top. They could be a biofilm on the side of a vat, or there could be fungal balls floating in the middle. So it's important to get a representative amount for that reason. Next, we're going to cover USP 62 testing, tests for specific organisms. The purpose of this test is to look for very specific organisms that may be present in the product, and this is a qualitative test, so we're just looking for growth or no growth of those specific organisms. So the testing is performed by using that same preparation that we described in USP 61 and placed in either broths after incubation or directly onto plates. So for example, a Staph aureus strain, the broth is placed onto a mannitol salt agar, which is selective for gram-positive organisms. But it's also differential, because if Staph aureus grows, as you can see, there's yellow colonies with yellow halos, whereas in the top right, that gram-positive, probably Staph epidermis, doesn't have the same characteristics. For Pseudomonas, as you can see, there usually are green colonies, and they kind of smell like grapes. For E. coli, that doesn't smell like grapes, but as you can see up in the top right, for coliforms, there's going to be purple colonies because they of their the way that they use the nutrients in the agar, whereas at the bottom they don't have that brick red color. And for Salmonella, again, you can see growth there. Since the screen is for Salmonella species, the growth may have different characteristics that are shown. Other screenings are outline testing, Candida albicans, Clostridia, and bile-tolerant gram-negatives. Once the test is run through and we get characteristic growth, confirmation tests are performed, for example, genetic ID to confirm that that's the actual organisms that was recovered. And back to growth motions, it's very important for these selective and differential broths and augers that growth motion is performed to ensure that the media is making doesn't allow incorrect organisms to grow and shows the right differential areas for the correct organisms. Limitations for this test, it's screened for only those specific organisms. So if you ran a E. coli test and you get growth, but it turns out not to be E. coli, at the second step there, it should be ID'd, because if the testing is able to pick up that organism, it's very likely that E. coli could also grow, and that's showing that there's some type of problem in the system. But if through a acceptance, if this acceptance criteria is set to have an organism that's not outlined in the USP, a separate screen needs to be created. Next, we'll talk about antimicrobial effectiveness testing. And as the name says, this is a test that tests the effectiveness of antimicrobial properties in samples to ensure that the preservatives are working. The testing has to be performed on sterile, non-sterile products that contain antimicrobial preservatives, even if it's not, if the active drug is naturally antimicrobial. For sterile products that are multi-dose, these contain preservatives that inhibit growth during withdrawal for individual doses. So if you get a shot, you have the vial with the rubber stopper that the syringe is going in and out. The preservatives are making sure that that is sterile each time. For non-sterile products, preservatives are used for to help control the growth, microbial growth during usage, and also manufacturing. The testing is performed on products as it's distributed by the manufacturer. And preservatives in the samples should only be at a level that's not toxic to the user. Preservatives are naturally toxic. That's how they don't allow organisms to grow. So you don't want to put so much in there that it could actually affect the person using it. In the USP, the testing is broken up into 4 categories based on route of administration and risk. So category 1 is sterile products, injections, sterile nasal, eye drops. Next are non-sterile products that are topical, anything that's included with mucous membrane. And then 3 and 4 are oral products. As we covered before, those are a lot lower risk. So during the testing, the inoculum is prepared. The organisms are grown per the USP requirements the same way every time. That helps ensure consistency during the test. We can't use the same exact organism cells. So to have reproducibility, the organisms have to be prepared exactly at the same time. The samples are inoculated with the organisms, and then a portion of that is plated at time 0, and then the preparation is incubated and plated at different time points. So example for category 1, the requirements are to have not less than a 1 log reduction from the initial calculated count, so from day 0 at day 7, at least a 3 log reduction at day 14, and no crease of organisms from 14 days to 28 days. And that ensures that preservatives are working. And then different requirements are for lesser risk products. Limitations for this test. Preservatives aren't a substitute for good manufacturing practicing, so you can't make samples in a garage and just add preservatives to make sure it's clean. GMPs need to be followed. The testing is focused only on the formulation and not the packaging, so it's best to understand the container closure properties of the sample. And as I covered, microbial variability, since you can't use the same cells every time, it's important that you have a microbiologist that knows how to do the testing so that you can ensure that the testing is done the same every time it's tested. And there's no suitability test included in the standard currently, but there is guidance in USP 1227. So we talked about testing. Next we want to talk about the requirement of objectionable in the intended use and to be free of objectionable organisms. USP 1111 is a guidance chapter that covers acceptance criteria for non-sterile products, and it's based on route of administration. So for example, cutaneous samples, products used on the skin, they recommend less than 200 CFU per gram or mil for the aerobic testing and less than 20 CFU for yeast and mold, and an absence of Staph aureus and Pseudomonas. And those are could be common organisms that would cause infections if the opportunity arises. It's opportunistic pathogens. But these are minimum requirements. Additional risk-based assessments should be performed, and we'll cover that here. For risk-based assessment to actually determine the acceptance criteria, the assessment should be performed by a microbiologist, someone who's familiar with the product, and would be able to identify key areas during the manufacturing and with the use of the product. You may need a medical expert for potential patient risks. So once you start the assessment, first you want to review critical control points, the route of administration, physical characteristics, and the intended user and how the product is used. So for route of administration, which we've discussed in USP 11, knows that site of application helps determine the risk of infection that's used on the skin or oral, nasal, and transdermal patches are going to have far different risks than a rectal use product. Next, looking at characteristics, does the product support growth? Is there a lot of water activity which would support growth? The pH, are there organisms that could grow in the sample at the pH or possibly change the pH, as I've listed there for acidic products contaminated with oxidative yeasts? And are the preservatives in the product appropriate for controlling the organisms that could be considered objectionable? Again, packaging, which we've talked about, and if the product has corticosteroids that can actually lead to the patient or the intended user being immune compromised. Finally, you want to look at the intended user and how the product's used. Is the user for healthy humans? Are they immune compromised? An example is cystic fibrosis. There's been a push by the FDA that we'll discuss in a moment for those immune compromised patients. If they're infants or chronic disease, or is it a target population, a mix of a lot of different statuses, where you may want to take the highest risk and base your risk assessment there. Method of application, is it possible the person will misuse the product? Example there, I have eye drops before container closure requirements were to have it into a bottle. They would have separate droppers that weren't actually a part of the container, which led to contamination. And if it's used in open wounds or present disease, most likely the product should be sterile. But again, as I said, we have tissue samples that are not sterile that we test, but they have far different requirements. So next, we want to use that information and identify the objectionable organisms. Again, that definition of objectionable organisms is not 100% clear, but generally you want to look for organisms that are specifically a risk for the user of the product and organisms that cause spoilage. So for the user, and how it's used, as we covered, USP 1111 outlines the requirements by Brown administration, but there was a paper done by Scott Sutton that looked at FDA recalls and showed that a lot of the recalls aren't due to the organisms that are actually listed in the objectionable or in the tests in USP 62 or the suggestions in USP 1111. For example, Burkholderia cepacia for the cystic fibrosis patients. If there's a contamination in the inhalants or mouthwash products that can be aerosolized and breathed in by those patients, they're extremely at a high risk for infection. Or oral products, Enterobacteriaceae is a common problem. In the past, there's been issues with alcohol pads contaminated with Bacillus cereus that cause deaths. It's important to stay up to date with trending and understand where the industry is going so you can stay a bit ahead for your products with safety. Next, looking at the risk to cause spoilage. Is it possible to look at what type of organisms could possibly change your product? Are there activities that could degrade the proteins? It's found that Pseudomonas contamination can decrease the active drug with some drugs, or even some organisms can inactivate the preservatives in the product. Another sign of spoilage is appearance, discoloration, different odor than normal. Top of products can become gritty. Oral syrups, since they have lots of sugar, yeast can cause fermentation. And when there's gas production, they can cause problems with the container. We've had a few samples in the laboratory that have exploded due to yeast contamination in the product. And you don't want that happening on the store shelf, obviously. So using that information, you want to start to determine acceptance criteria. Using 1111 is a good starting point, and then looking at those critical control points, using that information to identify hazards. But a further risk of management may need to be performed. For example, failure mold effectiveness and critical analysis, or hazard analysis critical control points for food that's used in the food industry is common in the pharmaceutical industry. And then risk management. Once you've had the acceptance criteria set, you always want to continually having corrective actions and documentation, having corrective actions when action levels are met, or looking at trending. Document everything. Honestly, if the FDA comes in, it's always best to have everything that's available. And if anything changes in the manufacturing, you will most likely need to do an additional assessment to see how that could affect the products. And having a microbial control strategy, identifying the sources of contamination, contamination control, monitoring raw materials, environment, finished products through the tests we've covered, and performing risk assessments. So just an overview. Again, we talked about product types, sterile versus non-sterile, the history and basics of pharmaceutical microbiology, microbiology control and risk management, non-sterile standards and testing, and acceptance criteria. I wanted to thank you for listening to the presentation. If you have any questions, my contact information is listed there. Thank you.